Hello, and welcome to the Behind the Lab series. Getting started, if you can just please introduce yourself by telling us your name, the center and institution that you're connected to, and how you're affiliated with the MMPC.
Sure. My name is Brian Finck. I'm a professor at Washington University in St. Louis at the School of Medicine. And I have been affiliated with the MMPC for several years in that I've been a user for more than a decade. We ship mice to them and they do exhaustive metabolic phenotyping of the mice. And then more recently, I've joined in as an advisor to them on their external advisory committee. And so I've been closely affiliated with them for quite some time now.
Amazing. What notable work have you done being a part of the MMPC and just being affiliated with the organization as a whole?
Sure. A couple of the projects that we have collaborated with them on include studies that we did where we sent them mice that had obesity, and we sent them a drug that we wanted the mice to be treated with, and they treated them for several weeks. And then they did hyperinsulinemic clamps, which are the gold standard for assessing insulin sensitivity and glucose metabolism. And so that project identified a novel drug that improved glucose homeostasis in mice. And we are continuing to pursue this drug as a potential treatment for type 2 diabetes. And then another thing that we've collaborated with the MMPC on is to characterize the effects of genetic knockout of certain proteins involved in mitochondrial metabolism to determine how that would affect the exercise performance of the mice. And so the MMPC took our genetically engineered mice and they characterized metabolism while the mice were running on a treadmill. It's an extremely sophisticated technique and something that very few places in the world can do. And it's very powerful for characterizing how different organs in the body respond to exercise and how they participate in that coordinated physiologic response that is required to maintain the workload of the muscle during exercise. And so both of those studies, we were very pleased with the results. We felt like the characterization of the mice was very thorough and complete. And it definitely helped us to publish that work in respectable and solid journals to have that rigorous characterization.
So good. Since using the services with MMPC and just doing the work that you've been doing thus far, have you received any major grants or awards during that 10-year time that you would like to highlight?
Sure. The MMPC was involved in several of the grants that we've received in the past 10 years or so. Usually what we do is we request a letter of support from the MMPC attesting that they can do the studies that we are proposing. And I think it benefits us in that our institution does not have a core that offers many of the services that the MMPC does. And so it goes over very well, in my opinion, with the grant review committee to know that we're going to be using experts to do these studies. So we've received multiple R01s where the MMPC has provided a letter of support prior to submission. And then once the studies actually commence, as I said, we ship mice to them or we purchase mice and have them shipped directly to the MMPC. And they do the studies that we proposed in the grant. So it has been very beneficial in helping us obtain funding to do these studies.
That's awesome. With that, because that time frame that you've been using the MMPC, have you on any of your either current projects or past projects been able to see any medical or research advancements that you've been able to kind of participate?
Sure. I could highlight the one thing that we have done. As I said, we shipped mice that were obese and treated with a novel drug that might have implication or might be a future treatment for diabetes. And one thing that the core told us, or we found from the studies that the core did, I should say, is that this drug did not really sensitize the mice to the effects of insulin. So they were not more insulin sensitive. but their ability to handle glucose was improved. And that's an important distinction because it helps us to understand exactly how the drug is working with its anti-diabetic effects and gives us clues as to the mechanism of action and which target tissues we might be interested in studying further, or because the drug would be having effects on certain tissues but not others. And so I think that the studies that the MMPC did gave us an important insight into future studies developing this drug as a potential way to treat type 2 diabetes.
Thank you so much for that insight. Even with that being said, what inspired you to work specifically in the space of type 2 diabetes and obesity? Was there any personal interest to this prior to doing this research, or is there anything that inspired you to do this?
Sure. There are certain people who always just have a love of metabolism. And I found that about myself, in that even from early days when I was a graduate student, even though my project was not directly related to metabolism, I was interested in potentially roping in metabolism into the scope of that project. I've just always been fascinated by the complexity of it and how intricately it's regulated by various organs and different intracellular mechanisms. But on a personal level, I do have, I have many family members who have obesity. I have many family members who have struggled with type 2 diabetes. And so that personally drove my interest in studying diabetes as a metabolic disease.
And I'm sure also the ability of or the knowledge of knowing that you are helping others essentially with your work, and the impact that it's going to make on the general public, I'm sure is probably also good to know in the work that you do.
I would definitely agree with that. My PhD studies were very mechanistic, very focused on one specific aspect. And it was always hard to, when I would see family members or people would ask me what I study, it was always hard to explain because it was so obscure and hard to relate to the general public. And so when I came to Washington University to do my postdoc, one of the things that attracted me here was that it was a major medical center, and I would be working with many physician scientists who deal with patients on a daily basis. And I felt like the work that I was doing could then inform them in their patient care. And I really wanted to make an impact on human health by coming to a place that does translational research, that has people who study both basic mechanisms of metabolism and also, who see patients and can translate that to actually improving people's lives.
Are there any papers that you are most proud of, or that even have been recently published that you would like to highlight for our audience?
Sure. The one that I wanted to highlight today, which is a direct result of our studies with the MMPC at Vanderbilt, was that we took mice with a genetic defect in mitochondrial metabolism that was specific to the liver. And most people don't think about the liver when they think about exercise, because they kind of focus on the skeletal muscle as the organ that's doing most of the work that is, driving people's movement. But the liver is also incredibly important for sustaining that work over time. And it's part of this cycle that was discovered many years ago called the Cori cycle. It was actually discovered here at Washington University. But the Cori cycle describes how the muscle and liver talk to each other during exercise. So during exercise, when the muscle uses glucose as an energy source, it produces a compound called lactate. And lactate goes into the blood. Lactate then goes to the liver, and the liver converts it back into new glucose that can go to the skeletal muscle and be used to drive mechanical work. The problem is, in our mice, that we had this genetic defect in mitochondrial metabolism. They could not convert the lactate back to glucose. And so this cycle, the Cori cycle, was disrupted. And we found that the mice could not exercise at the same level as the wild type mice. And so that paper was published about a year and a half ago, or almost 2 years ago. And I was very interested in that whole concept and whether we could disrupt the Cori cycle and have an impact on exercise. And I think that's one of my favorite papers in the last few years that we've published.
Thank you so much for sharing all of this incredible information. And I'm sure our audience is just going to be grateful to just hear your insight on MMPC your experience and just the work that you all are doing over there at Washington University as well. Just have a few more questions. What are, in your experience over the last 10 years, what are the top 3 things that you think that everyone should know about the MMPC, just based on your work with them and your knowledge of them?
Sure. I think the top things are that it's a center that does extremely rigorous metabolic phenotyping. There's few places like it around the world, and they can do things that are beyond the capabilities of most individual labs. I would say the other thing is that they're incredibly willing to collaborate. They're willing to, to set up the agreement however you like. It can be a collaboration where they're considered authors on the final work, or they are just doing this on a fee-for-service basis. And then lastly, it's very easy logistically to get your mice to Vanderbilt. They have all the systems in place to transfer the mice. They have the proper IACUC approval. It's very easy and seamless to get these things done. And so those are the top 3 things that I would like to convey.
Thank you. And would you also say that your experience as far as the collaboration has been a positive one, or just have you had much experience as far as collaborating with other centers as far as them having information on maybe even a test or anything that you guys can utilize at your university that you've experienced?
Sure. They're very when the data are analyzed, they're very willing to sit down with you by Zoom meeting and discuss the interpretation of the data, give advice about whether the experiment needs more mice or whether you need a different analysis. And they also, are willing to share other general things that you could do at your own institution. So for instance, Julio and Luis are willing, to meet with you and provide insight into things that you could do in your own institution and the proper way to do them, the most rigorous way to do them. And so, they can convey that information by Zoom, and you can do some of the things yourself. But they're always there for the more invasive and sophisticated and complicated assays that you might want to do.
That's so good. So good. The final question that I have is, if you could tell your younger self who's starting out in academia, starting out in research, anything, if you could offer up any piece of advice, what would it be?
Oh, that's a tough question. I guess I would say that it's all going to work out. That that the hard work is going to pay off, and that things will come to fruition. Your projects will eventually work out one way or the other, and not to sweat all the small stuff and the small worries that are always popping up in the minds of graduate students and postdocs.
This is awesome, and I'm sure that the young researchers and investigators who are coming up would definitely appreciate this advice as well. Thank you so much, Dr. Finck, for all of your insight, wisdom, and just your experience with the MMPC. And that concludes our Behind the Lab.